Good evening to all the participants and good evening to all the audience who have joined in for this particular session. A very warm welcome to another session of COVID webinar series brought to you by Mitra and Company. I'm your host, Dr. Sharbhanu Ghosh, and I am the Customer Relations Manager at Mitra and Company. Today, we are presenting this webinar on the diagnosis of COVID-19, and our distinguished speaker today is Dr. Ashok Rattan. A very good evening to you, sir. And it's a pleasure to have you again today, sir. So Dr. Ashok Rattan will be speaking on the diagnosis of COVID-19, and his mantra will be that one size doesn't fit all. And before we begin with the formalities of this particular session, just to apprise to all of you that being a technical seminar, I presume everybody would have some questions to Dr. Rattan at the end of the session. So I would request all of you to go through the presentation as it proceeds on, and you can put your questions in the chat box and we will take it up at the end of the session. And after this particular session is completed, the recording of this will be uplinked into the YouTube, so you can always have a recording over there also. To begin with, just to apprise, as we have been constantly engaging with the technical fraternity and taking awareness with respect to all the parameters so as to speak, we have partnered with TIDE, which is an initiative of Mitra and Company. So to begin with, I just want to give you a short introduction about TIDE also. So TIDE is the annual feature of Mitra and Company, and it is about the technologically domain where we discuss all about the various platforms and all about the various upgrades which is brought about in the diagnostic industry. This is the web page of TIDE 2021, and it's going to be conducted from 1st to 3rd of December. And it is a platform which focuses on the developments of the impact of the diagnostic industry, and it discusses and argues all about the policy matters, the emerging trends, the opportunities which are there, the new research which has taken into effect, and also discusses the roadblocks and the bottlenecks which we encounter in the daily routine. It will address the stakeholders and the specialists of the industry, be it from the in vitro diagnostics or POCT, molecular diagnostics, and even from the research and development sector. The broader aim of TIDE is to catalyze the revolutionary changes in the Indian diagnostic space. And we are just providing that platform over here so that everybody can share their opinion in a more accessible and affordable diagnostic approaches which can be brought into the market. And we just take forward the government's key role of mission of Swasth Bharat and Shresth Bharat. Now, the speakers of TIDE are constantly coming on board. This is just a snapshot of the last year's event where we had distinguished speakers all the way from Malaysia and across the globe. So this time we do have an editorial board, and the engagements will be there in the form of webinars. There are case studies where the speakers can present their case studies, and also the publications which they have published till date. And they are also free to put their blogs. So I request all the experts who are joining in today that they can feel free to contact us, and we will be more than happy to welcome them on board in the editorial board over here. The website of which is already mentioned over here, and you can get all the details in this particular website. Furthermore, there will be CME sessions also on 3rd of December, and the webinars will be on the topics of COVID-19, hepatitis B, diabetes, and celiac disease. So this is exactly will be the agenda for 3rd of December. Furthermore, just to apprise you that this particular CME session is free of cost. We are charging nothing for this particular session. So please feel free to come on board for this particular session. Also, we are engaging with various platforms, and we are reaching out to the common mass. So we expect the posters also to come in. So there will be an online poster submission also. And this is also free of cost. So all the details have been put onto the website. So the categories will be in the form of letter to editors. It can be case studies. It can be a review article, or it can be a novel research article also, which is published. So we would welcome in any format which you put in over there. And last but not the least, whatever the engagements we do over 3 days in this particular conference will be recorded, and there will be a live feed also, and it will be uploaded in YouTube. This is just a snapshot of all the videos which are there currently from last year's event in YouTube. So I presume that everybody can have a wider interaction and can engage with various opinion of yours in this particular platform. And I wholeheartedly welcome all of you to come on board for this TIDE 2021 series. So moving on with this particular thing, I welcome Dr. Ashok Rattan for today's webinar. And Dr. Rattan has been at the forefront of bringing all the new technologies and assays for COVID-19. And equally, he has been arguing a lot about the newer technologies which should be brought on board. So over to you, Dr. Rattan. The house is yours.
Thank you. So you'll activate screen share?
I have already done that, sir.
Okay. So good evening, everybody. As you know, that on 31st December 2019, China informed WHO regarding outbreak of pneumonia caused by novel coronavirus in Wuhan. And by the 10th of January 2020, one full sequence of this RNA virus was made available in the public domain. In the next day itself, 5 more full sequences of this RNA virus became available, and researchers both in Berlin and in CDC Atlanta identified that this was a novel coronavirus, unlike the previous 2 systemic disease-causing coronaviruses, and that there were specific genes which could be identified in the N region and the RDRP region of the virus. And so they quickly devised and started using RT-PCR because this was an RNA virus. Of course, that has not stopped many celebrity doctors from saying what needs to be done and what should be done and what is being done. But WHO said that since this is a new virus and all of us have no experience with it, what we should do is test, trace the person who's infected, and isolate the person so that the spread doesn't occur. It is to ICMR's credit that in January 2020, when the virus first came to our notice, many of us had no idea about coronavirus, and only one lab in National Institute of Virology in Pune did have the primers to do RT-PCR gifted by the WHO. But by September 2020, thousands of labs have been commissioned have been permitted to test for this virus. And worldwide, there has been an avalanche of tests available. Many of them are molecular, some of them are antigen detection, and still some are serology. But as you see here, 1,122 tests are in various stages of development. Many of them have been approved by regulatory authorities. WHO has been the most stringent, and so they have permitted only about 22. 20 of them are molecular and 2 are antigen detection. But many other authorities have permitted even US FDA, which is normally very stringent, has permitted hundreds of EUA applications. If we take a step back, I looked through my slides and I saw that on this day last year, I was addressing the biotechnology students of Vellore Institute of Technology. And I told them that this virus was the third coronavirus which has caused systemic infection. Before SARS-CoV-2 came SARS-CoV, this was in 2003, which caused about 8,000 odd infections, some 774 deaths, and then within 6 months, it disappeared. Then you had, after a decade or so, in 2012, in Saudi Arabia and Jordan, were Middle East respiratory syndrome virus, that's MERS-CoV, and that has been smoldering because people there have been able to take good control of it, and it was only when it got transmitted to South Korea that an outbreak occurred in South Korea. And now in 2019, we have SARS-CoV-2. So while the other two viruses have limited spread, in July, when I was speaking to them from March, where there were 2 million cases, by that time, 6 million cases had occurred. And then on this topic of one size doesn't fit all, I had spoken with Dr. K.K. Agarwal on 19th February 2021, and at that time, 109 million cases have occurred. And today, when I speak to you, 184 million cases have occurred in the world with 4 million deaths. So you would agree that whatever we are doing is not working. When you look at Government of India's website, there you find they have given thousands and thousands of vaccination. They are testing thousands and thousands of samples, but the cases just keep coming. So if we take a step back and try to understand why SARS-CoV disappeared in 6 months' time, MERS have been smoldering in the Middle East, why SARS-CoV-2 has spread as a pandemic. One of the reasons would be that this virus multiplies in the upper respiratory tract rather than only in the lower respiratory tract. The second one is that asymptomatic or pre-symptomatics also transmit the infection. Unlike SARS-CoV and MERS, where persons who are symptomatic will transmit infection. And this we knew by May 28 last year, around the same time that FIR was being filed against one of the hospitals in Delhi for testing asymptomatics. That was the time when in USA, they identified that asymptomatic transmission may be the Achilles heel of current strategies to control COVID-19 infection. So if we summarize what we knew by the 9th of July last year and what we know after 1 year, that this is a new virus. Now, whether it has been manufactured in the lab, or it is through natural selection, it's still up for debate. It has caused an unprecedented pandemic in modern times. There is no proven therapy. That was in July 2020. There's no preventive vaccination. That was in July 2020. Now we have vaccination. So there's been correction from that point of view. That proven therapy, we now know that steroids will work, and so would monoclonal antibodies. And there is no proven vaccination. Instead of that, now many vaccines are available, and most of them are effective. We are definitely in uncharted territory. Diagnosis is proving to be an important tool to inform patient management and potentially help in saving lives by limiting spread of infection. But diagnosis alone would not do it. We need to trace and we need to isolate. What to test, whom to test, and when to test, ICMR has been advising us repeatedly, and I think they've given 9 or 10 guidance on what to do. What has not been very clear and whose responsibility it is, what to do when a test becomes positive. And that is why we've tied ourselves in knots. We also know that this infection could be divided into 3 stages. In the first few days is the infectious stage, where the virus is in plenty. Thereafter, it goes into the lungs. So there's the pneumonia stage. The virus becomes scanty and sometimes disappear. And finally, some people from pneumonia would go in for inflammatory phase, where there's cytokine strong, and that might end fatally. We also now know that if a person is to survive, he must have neutralizing antibodies by the 14th day. If there are neutralizing antibodies by the 14th day, study in Yale University in USA has confirmed that a person will live if neutralizing antibodies are present as a consequence of infection or after vaccination. WHO has also indicated that you might want to do testing for surveillance, for case management, for contact tracing, for monitoring of response or recovery, for prognosis, for vaccine response, and for environmental monitoring. And different tests should be used or should be considered. This naturally comes to pass that if we talk only of RT-PCR, then definitely one test is not fitting all situations. We should ask ourselves relevant questions, and we should then find out the test which is most suitable to address that question. Some of the questions says, is the person infected and should he be isolated? Is he infectious? Has the person recovered? Can the person be safely discharged? Is the person immune to reinfection? Can he donate his plasma? Has this person been exposed? Does he have protection against infection? Has he responded to the vaccine? Is this reinfection? Has the virus mutated? And is this a new variant? If these are some of the questions that come to your mind, the need is to identify infected persons and isolate him as soon as possible, which unfortunately is not happening unless there is a lockdown. To hold him in isolation as long as infectious, to avoid admission, to advise admission in hospital or ICU as per severity, to discharge him from isolation when no longer infectious. Is this person suffering from asymptomatic infection and should not travel? Has this person been exposed, which would be through serosurveys? Does this person have protection? Does this person respond adequately to vaccination? And I would tell you that not everybody is responding to vaccination. Is this case a case of reinfection? Is this a new mutant? And let me share my views on this. The first part was about the sample. There are various samples which could be used, and unfortunately, the ones that have been chosen are the nasal swab and the throat swab. There was already a publication in JAMA in March 11, 2020, from results of testing in China, which said that nasal swab will give you 63% positivity, while pharyngeal swab will give you 32%. And we have, of course, the infection is through respiratory route, and so respiratory secretion is what is required. And for whatever reason, we have chosen nasal swab and pharyngeal swab. This is not nasopharyngeal swab, because I'm sure many persons have no idea how to collect nasopharyngeal swab. So it's either the anterior turbinate, the nasal swab and pharyngeal swab, throat swab that we're using. But Yale University had already indicated sometimes last year, this was April 22nd, 2020, that saliva is more sensitive for SARS-CoV-2 detection in COVID-19 patients than nasopharyngeal swabs. The viral load in saliva was higher and persistent and consistent, while nasopharyngeal swabs one day could be positive, the next day negative, then again positive. So that's one of the reasons for tying ourselves in knots. The second, of course, the good point has been that we have had RT-PCR, and what is recommended is that multi-gene RT-PCR should be used. Some people have looked at multiplex PCR, where since the symptoms are similar, they might want to look at influenza A, H1N1, H3N2, and B along with COVID. Then they looked at point of care testing so that the results can be returned faster than antigen detection. Antigen detection will always be inferior than RT-PCR, because in RT-PCR, you're amplifying, using PCR, you're amplifying whatever the RNA is present 1,000-fold or more than 1,000-fold, while in antigen detection, you're only fragmenting whatever is the virus, and then you look at that. So that will always be inferior and less sensitive than RT-PCR to start with. So direct demonstration always is better to make a direct diagnosis, but we can also look at host response, and I will show you some results of the tests that have been done, that host response could be as an adjunct to making a diagnosis in acute infection after the first 9 days are gone, because antibodies first start appearing about 7 to 8 days. So before 9 days, it's worthless testing for antibodies. But neutralizing antibodies can, if you are wanting to give monoclonal antibodies, then as soon as the patient becomes positive, you might want to test for neutralizing antibodies, because if neutralizing antibodies are present, then monoclonal antibodies would not be of any use except for the cost. But if they are absent, then monoclonal antibodies can be life-saving and prevent the progression of a person who's RT-PCR positive with comorbidities from suffering from severe infection. There's also been suggestions that instead of only antibodies, we should look at cell-mediated immunity. and at least 2 kits are available. Not in India yet, but in the Western world, people have started looking at CD4 and CD8 as well as interferon gamma. And of course, everybody is looking at sequencing for mutation. Normally, people talk about next generation sequencing, but from Britain's experience, it appears that multiplex PCR, since mutations of consequence are only in the spike gene, then you might want to use multiplex PCR where you can identify the changes at site. So as I was saying, that in any diagnosis, you try to look at RNA in the first 2 weeks, and after the first week, then you look at the immune response. The nucleic acid tests which have been used for COVID are mostly RT-PCR, but TMA, RT-PCR, and CRISPR, RT-RAMP and CRISPR have also been successfully used. As I indicated, since this virus is known to mutate very frequently, it is always better to have multiple genes and various kits, either manual or automated, have been devised to looking at various genes. We also know that when Nelly variant came in US and UK, which is now known as alpha variant, there was a mutation, there was deletion of 6970 position, which is the site where the S gene binds. As you see here, if you are using Thermo Fisher's TAGPath multiplex PCR, then you would give positive results only when there is N gene is detected, S gene is detected, and ORF1ab gene. At least 2 of these 3 genes have to be detected. And when mutations occurred, alpha variant N501Y had deletion, and that caused the S gene silencing. Then S gene silencing did not make any difference in detecting. In fact, instead of sequencing, they started looking at S gene, and they started saying that this is the mutation, this is the alpha variant that has occurred. And they could make, instead of sequencing everything, they could make it, they could do epidemiology of this variant, looking at S gene silencing. It was in Wuhan itself that Lee had indicated that RT-PCR, which somehow people have been saying is the gold standard, at best of time, it was 80 to 90%. In the first week, 80 to 90% of cases which were clinically suspected to be COVID were detected. And it was only when they added serology that after 9 days, you would be able to get more cases as there's decline in the positive predictive value of RT-PCR. Gao had also indicated that if you use RT-PCR alone for making a diagnosis from clinically confirmed cases, you would have less success. If you add serology to your diagnosis, then you would be able to, of course, serology kicks in after 9 days, and this was made very amply clear by Miller in Boston, who had the policy of using both RT-PCR and serology on all persons admitted in the hospital in Boston. And then they found that once you, in Harvard, when you admit all the patients and you test for both serology and RT-PCR, by the 9th day, as the RT-PCR keeps decreasing, the chances of serology adding to your diagnosis ability keeps increasing. And by second and third week, nearly all patients had serological evidence of infection, while RT-PCR would keep decreasing. If you're looking for gold standard, then gold standard for microbiology is normally culture. And when you do for virology, it is a cytopathogenic effect on cell lines. And coronavirus grow very well in virus cell lines and produce cytopathogenic effect, as you see here, as the holes in the monolayer, the cells will die and they would not then be stained. And it was Bullard who could do both RT-PCR and culture, who indicated that when and he was in Canada, that from Canada he indicated that when as long as CT value was less than 24, the sample would have a replicative virus. If the samples, if the CT value became more than 24, then they would be dead virus, or it will be a remnant of RNA. So there is no infection. It is not infectious after the CT is more than 24. When it was discussed in the meantime, since PCR was taking too long, two instruments, Gene Expert and TrueNat, which normally were available in plenty in various labs in India and were used for tuberculosis, the manufacturers could devise them for SARS and make the cartridges available, or the lateral flow cartridge available, and they started using it for tuberculosis. Instead of tuberculosis, they started using it for COVID. Though the same persons who were confident in using Gene Expert for tuberculosis, when they started using COVID, had objection to use and to giving out CT values, but they had to be reminded that when they were using Gene Expert for tuberculosis, where CT value had no value, the instrument will still give CT values. So while there was some evidence that CT value in coronavirus would indicate whether a person is infectious or not infectious, and Pooja Das from New York had reported that if at the time of admission the viral load was very high, then the chances of fatality were more if you follow up the patient.
So for the COVID-19 test, we've stopped using VTM and we've started just using sterile tubes, and we've been collecting nasopharyngeal and oropharyngeal specimens for each COVID-19 test that we've been running.
So since RT-PCR was taking too long, then ID now became available, and Thermo Fisher recently has introduced Aquila, which again give RT-PCR in 30 minutes flat. You have the sample and you load it into that, and in 30 minutes you get the result. So, but RT-PCR and the role of RT-PCR has been under question. Meena, again from Harvard, indicated that RT-PCR, as I tried to indicate, that at best of time it's 80%, but there would be sometimes it is more sensitive during the initial stages and will give you false positive results. And since it will amplify leftover of RNA, it will amplify whatever RNA is left and could be positive for weeks together, when we know that the virus multiplies or is replicative for 7 days to 8 days. 8 days is maximum, and that is why once everybody understood that replicative virus is present only for 8 days after symptoms appear. Then for discharge, initial was to get to RT-PCR negative. Then they decided that as the viral load is declining and is clearing, if 10 days are passed after appearance of symptoms and the last 3 days are without any symptoms and fever, then a person can be discharged and he is not infectious. So CT value played an important role in defining those who were infectious and those who were not infectious. Then there were points raised that RT-PCR may be too sensitive, and that is where the antigen detection became available. And the aim of antigen detection was that since it will detect the presence of virus, which will be fragmented, and you will test the presence of nucleocapsid protein, the most prominent antigen of the virus, this will be less sensitive than RT-PCR. The virus, the test itself, to start with, will detect only the presence of antigen. And since it will detect only the amount that is already present, and there is no amount of amplification required, as a consequence, it will be less sensitive than RT-PCR and would not mislead you in keeping a non-infectious patient in isolation for longer than what is required. The only trouble is that antigen detection is not only less sensitive, it is highly less sensitive than RT-PCR. And so even persons who have less than 24 CT value may still be reported as negative. So the aim was that RT-PCR would detect very early, and it continues to detect for weeks together. The antigen detection would detect when the virus is present and may be more relevant, but it seems to be inferior to detecting infectious virus alone. So what is then required is some amount of amplification, but with the results available in 30 minutes time, so that a person who's positive could be isolated at once, which is the cornerstone of blocking the spread of this pandemic. In this case, the reaction is in one tube and everything, and there are multiple primers, internal primers and external primers, and a loop primer, so about 3 sets of primer, and the amount of DNA synthesized is tremendous, so that in 30 minutes time, you would be able to get the results, which can be seen by naked eye. It is 100% specific, accuracy is 99.35%, and sensitivity is 98.7%. And the beauty of this is that it requires only a thermal block, so that it requires is isothermic, so that only one temperature is required, 62 degrees Celsius. And this has been made by Agapi, which is a company, which is an Indian company in Cochin. And this has been validated by ICMR and approved. There is another local company in Gurgaon, which have used this mini dry bath, and they can get the results in 20 minutes time. And you can see the results. The positive ones are yellow, the red ones are negative. And if you want better discrimination, then you can use a UV light handheld system where the fluorescence will become yellow and the negative ones will become clearer. And they have been also validated, and they've got excellent results with RT-PCR.
Scientists are using a similar idea to test patient samples for novel COVID-19 RNA. To do this, CRISPR proteins are added to the sample. The scientists give the CRISPR proteins directions to identify the coronavirus RNA sequence, and then they arm them with a molecular alert system. When the CRISPR protein finds the COVID-19 RNA, it cuts the strand. Then this triggers it to cut any nearby molecular alert RNAs, releasing light. As time goes by, more alert RNAs are cut, and the light becomes brighter. The glowing light signals a positive result.
So overall, it will be that instead of RT-PCR, which is too sensitive and gives positive reaction much beyond the infectious stage, a test which is slightly less sensitive than RT-PCR, but definitely more sensitive than rapid antigen test like RT-LAMP would be ideal, because it will give reports in 20 minutes time, and you can then clinically isolate the person. We also know that the laboratories have been asked not only to do the diagnostic part, but also help in contact tracing, epidemiology, and surveillance testing. And definitely, it's not the same test which could be used for all. The screening test has different requirements than the diagnostic test. We've also seen that flights to Australia as well as Hong Kong have attracted adverse comments, because when persons carrying negative report from India land in Australia or Hong Kong, they've been found to be positive. I am mindful of the situation that there are fake COVID tests which have been, so the testing has not been done, but a report has been given. But there was a paper which was published in September from China, which indicated that asymptomatics are not only those which are NAT positive without symptoms, but when they analyzed a large number and they use IgM detection, they found that a large number of those which were asymptomatic and infectious had not only NAT positive, but they also had IgM. So they recommended that if you wanted to identify, if your purpose is to identify asymptomatics and recently infected persons, then you should look at not only nucleic acid testing, but also IgM testing. And if either of them is positive, you should prevent them from traveling, because they would be infectious and they would infect others. Now, when you want to do serology, you should be mindful of what antigen you're using and what method you're using. Initial step was to use lateral flow, which is not sensitive enough. The advantage of lateral flow is it can use finger prick blood, and it gives you results in 10 minutes time or 20 minutes time. But it is not sensitive, and for diagnostic purpose, it's of no use. You should either use an ELISA test, which is made automated by using a washer, or fully automated by using a DS2 instrument, or use chemiluminescence, which is fully automated, because this will give you better reproducibility. The advantage of ELISA is it can be carried out in any adequately equipped lab, while Lysin or chemiluminescence would be in a central lab. And so you should look at what is the antigen that you're using. And if you're using nucleocapsid protein, since nucleocapsid protein is inside the virus, it can have no role in protection. On the other hand, it is the spike protein which binds to the ACE2 receptors, but not all antibodies which bind to spike protein will be protective. It's only those which bind to the RBD would be protective. It's like a person with a knife entering into the room. If you can block his hand, which carries the knife, you have neutralized him. If, on the other hand, you hold any other part of the body, he can still attack you, and he's not. So you have bound him, but you have not neutralized him. This is an illustration of what we want to do. The infection starts with the virus binding to the ACE2 receptors. Our aim is that antibodies will bind to the receptor binding protein, so that now the receptor binding protein is completely saturated and it will not be able to bind to ACE2 receptor and enter into the cell. The best way to find this out is by doing a neutralization test, but this requires live virus. And since live virus is dangerous and you need PSL3 facilities, it's not suitable for diagnostic lab. This has been overcome by making a pseudo virus, and many tests have been described in China, protocols published in Nature, and in Italy also a simplified test available, so that this can be done in clinical labs, but it'll take 3 to 5 days. On the other hand, a new test which is genetically engineered, RBD and ACE2 receptors. ACE2 receptors are used to coat the ELISA plate, while RBD is what you're trying to neutralize. The antibodies, if they are present in the blood, will neutralize RBD, so RBD will not be able to bind to ACE2 receptor, and you can detect by ELISA. So this is a cell-free, virus-free ELISA system, which is completely safe and can be used in any adequately equipped lab. Initially, the test was devised outside India, and it was approved by US FDA, and now it's available. Both that test is available, and Mitra has also made a similar test, which has been approved by ICMR and DCGI. And as you notice on the right, the performance of CPAS, the internationally available, as well as the Mitra test, has a nearly equivalent result. It's also been seen, what is the use of finding this out is, if you're already infected, then one dose of vaccine will give you high titers. So maybe you don't need the second dose. But it's also been seen that not everybody, after vaccination, not everybody produces high titers. And if a person is over 65, then chances of low titers, both anti-spike or neutralizing, are there. As you notice here, that persons who are over 65 produce a less amount of antibodies, and they became susceptible. In animal experiments, it's indicated that 400 anti-spike, 100 RBD, or neutralizing antibodies of 50 are protective. Why some people die and why others survive has been addressed by this paper. It indicates that if a person by 14th day of infection does not have neutralizing antibodies, then that infection might end fatally. This is indicating AstraZeneca and how it will be able to address the Delta variant. And they notice that the effectivity of the immunogenicity of AstraZeneca is half of that of Moderna, and it is compromised again when you have variants. And so initially it was thought that one dose of AstraZeneca would give you good protection, which it did. It offers good protection against Wuhan isolate. but against Delta and other variants, it is better that you become fully vaccinated. So you should complete your full vaccination with Covishield or Covaxin as soon as possible, because 2 doses will give you better protection. And today also there was discussion that maybe a booster would be required further to boost your immunity. It is not to say that only antibodies protect. T cells will have an important role, but at this moment, though 2 kits are available, we are not aware of what is the threshold. We now know that Delta variant is spreading all over the world, and in America as well as in Indonesia, that has become the dominant variant which is causing infection. And these are the wheel which says what amino acid changes and takes the and makes the variant. And the mutations in other parts other than spike is mutations of no consequence. It is when mutation occurs in RBD that changes occur and immune escape can occur. To detect the variant, a number of RT-PCRs, multiplex RT-PCRs are made available, and Europe seems to be using it much more, though in India, everybody seems talking about Thermo Fisher or Illumina as next generation sequencing, which is both costly and maybe not required. Because if you're looking at mutations of spike protein, then a multiplex will do. To go to the other extreme, from Paris, a Paris score was devised looking at only CBC, complete blood picture, and giving different probabilities. And they gave different points to eosinophil levels, lymphocytes, neutrophils, and basophils, and said that if the Paris score was 1, the sensitivity was 100%, and you could then pick up, you can say that a person is not likely to have COVID infection. While if the score was 4 and 5, then if the score was 4 or 5, then the sensitivity specificity was 97% of finding a person with COVID infection. One of my friends in Saudi Arabia has devised a system where my values, where I've been COVID negative and a positive person, I compared the values, and when we entered the results, you would find that you enter the results, if the value is 1, there's low possibility, this is my results, and the patient's results when they were entered, there was 5, that means it was high probability of being RT-PCR positive. This could be tried out by persons who are far away from RT-PCR or from testing labs, and just on the basis of CBC, you can then guess whether the person is likely to be positive or negative, suffering from COVID infection or not. You also know that in UAE, the dogs have been able to send out COVID infections, and in Abu Dhabi, they've started using a handheld machine to look at the face and to identify persons coming into the mall, whether they have COVID or not. Artificial intelligence can look at your sound of the cough, as well as there are systems available where breaths which have volatile organic compounds can be analyzed and identified. So there are various ways to test, but you should use the test which best suits the purpose that you're testing for. And the questions that need to be addressed is, these are my suggestions of what tests would be used to answer the questions that we asked in the initial stages. But I think the time when we would collect hundreds of thousands of samples, take it to a central lab, and give report whenever we want to. And normally after 24 hours is over, and we need to now make more patient-centric approach with results available in less than 1 hour's time. And there are multiple tests now available which will give the results in less than 1 hour. And we should use them. And I'm very happy to note that my lab has started a self-test, which unfortunately is a rapid antigen test, which should not be as sensitive as you require. But the advantage is that you can test it yourself and you can isolate, because you can contribute by, if you are infectious, then you stay at home. Do not mingle with persons. But in India, our deviation from COVID appropriate behavior is currently the biggest variant of concern. I thank you for your attention, and I hope that you stay positive, but test negative for RT-PCR or any other test that you do, and have protective neutralizing antibodies after vaccination.
Well, I mean to say, sir, nobody would doubt that. Everybody should agree with that concluding statement. It was wonderfully placed and explained webinar, sir. There are a few questions which are lined up, sir. So I will take one by one. Dr. Bohra has asked that what decides the R0 value for a disease, and is the R0 value for SARS, MERS, and SARS-CoV-2 is the same, or is it different?
See, R0 will depend upon what is the possibility of spreading from one person to another. Even in Wuhan, even in COVID infection, Wuhan had, say, R0 of 3. Then you have D614G. That had much higher predilection of spreading. So these keep changing. And since the fit to ACE2 receptor was better, it could spread even more. And now, as you notice, that Delta is replacing everything else, because Delta has a better R0. That would also indicate that we should forget about thinking that that herd immunity will safeguard us, because herd immunity keeps changing and the virus is finding ways of escape, immune escape. Whenever it is immune escape, then even if you are vaccinated, you get infected. The advantage of vaccination is that you will be protected from severe infection and death. Very few deaths have occurred once a person is infected, once a person is vaccinated, but infection still occurs. So the best bet that we have is that we get ourselves vaccinated as soon as possible, but at the same time wear mask when in public, and have appropriate COVID prevention behavior consistently used.
Sir, another doctor is asking that since the Delta variants are coming up and the new mutations are in place already, they are bypassing the vaccine. So how can we be safe in this kind of a scenario?
Delta variant has mutations in the RBD. As a consequence, the antibodies are less effective. But at the same time, as I've tried to indicate, that it is more than only the RBD, maybe the T cells are also protecting. So those, and the antibodies also still, though the activity of antibodies have decreased, but is still higher than if a person was not vaccinated. So what is required is you should be fully vaccinated. That means complete your 2 shots as soon as possible and have COVID appropriate behavior. Even if COVID infection, that means Delta infection or Delta plus is not as infectious as Delta. And that is why Delta has spread all over the world. Delta plus is still localized in Kerala and in Maharashtra.
So Dr. Manish Pathak is asking that which combination of gene should be preferred for RT-PCR? Should it be the ORF-NS or it should be the ERDRP and ORF?
See, you should avoid S, because S, as I indicated, is the mutations in S are the ones which will lead to, which are having the immune escape. Normally, they have looked at conserved regions which are not subjected to mutations, and they do blast every time, so that they see whether the site where the primers bind, that is not being mutated. That is an ongoing exercise. But one rule of thumb would be you should have multiple genes that you're detecting, so that even if one mutation makes one gene silent, there are other genes which are positive, which will, so that you do not miss the diagnosis of COVID infection.
So there is a follow-up question to that. Like, if the CT value of 24 for swab samples may not be indicative, as it may depend upon the sample collection, so will it be the right thing or the wrong one?
Those stories are going on all the time. No? Even for tuberculosis, it had no value. So who's telling, if you don't collect the sample properly, then which result will be reliable? Then no result will be reliable.
Exactly.
So ultimately, instead of taking a sample which is likely to be patchy, the best was to have a gargle, a sputum examination or gargle, because the virus in the saliva is persistently and consistently of higher titer. But somehow we don't seem to be able to get it. It doesn't seem to appeal to us that instead of opening and putting a swab into the nose and touching whatever, you get better results. But if you get saliva, you will not get good results.
Sir, there is one question over here. Like, they are asking, like, which would be the ultimately the gold standard test to identify COVID? Will it be the RT-PCR or rapid test?
No, rapid test is not sensitive enough. So what you require is something like a molecular test. It will always be sensitive, but RT-PCR is more sensitive than you require. As you noticed that, see, in microbiology, it is normally culture which is taken as the gold standard. Unfortunately, we cannot do culture in this because we don't have BSL-3 lab. So those who have BSL-3 lab, they should continue to culture, and they are telling us, and there are multiple authors who have indicated that the virus from infection to is infectious for the first 8 days. After 8 days, in biology, nothing is 100%. But in majority of cases, after 8 days, the virus is no longer replicative. So if you have a test which is detecting beyond 8 days, then that is false positive. Because your purpose is not to detect the RNA, but to detect replicative virus, which can cause infection in others. And that is why a test like RT lab, which is likely to meet that requirement and give you results so that you can act. If it is positive, you have to act at once. You cannot isolate a person after 24 hours and hope that now the transmission is blocked.
Sir, there is one last question for you. Is it advisable to do neutralizing antibody detection even if if it is negative, and another booster dose is advisable?
But unless you do, how will you know that it's negative? See, at this moment, you are depending on belief. I must admit that no authority, whether CDC, FDA, WHO, or ICMR, is indicating that you should test your antibodies, right? But at the same time, every researcher, when they want to say that the vaccines are working, they are testing antibodies. And when you say that this variant will work or that variant will not work, is on the basis of antibodies. And monoclonal antibodies are now available at high cost, and they have been shown to protect if given within 72 hours of appearance of symptoms. So there are three places where you should test for neutralizing antibody. Is one, if you are infected, then you want to know by 14th day, have you got neutralizing antibodies or not? Because those who have survived have neutralizing antibodies. So on 14th day, if you are RT-PCR positive, get your neutralizing antibody done. And if you are positive, then you will survive. Second, if you are vaccinated, after 2 weeks of the second dose, you want to know, have you produced neutralizing antibodies? And third, if you are RT-PCR positive, before you use monoclonal antibodies, you want to see whether you already have neutralizing antibodies. If you have neutralizing antibodies, monoclonal antibodies are not required. So you can save your 50, 60,000 that you would be using for monoclonal antibodies.
So I think definitely a lot of avenues are still unexplored, like what you have said, sir. And we must all admit that since the inception of this pandemic, a lot has been done and a lot more needs to be done also. The assays have also been refined in the due process. And yes, what you have also mentioned that sampling error can be possible at any particular test. So definitely we have to take precautions while we are sampling it out. And the best thing is that, yes, we have to get the vaccines at the earliest. So there is no alternative to it. And we cannot drop the COVID appropriate behavior. Unfortunately, a lot of people are not taking it seriously. So
I don't understand why they don't understand this, that there is no escape.
yeah. So we have been getting very disturbing images from all the tourist places, and that's a very harsh reality that people are still taking it very lightly. And I request all of you to keep on making the awareness that it should not be taken lightly. Well, concluding this particular sessions, a small piece of appreciation from our end. So this is your certificate of appreciation. I constantly thank you enough for educating people like me even. So I really look forward to hear from you again in the near future, sir. And it has been a very interactive and a very enlightening session, sir. And to all the participants over here, just to apprise you that we will constantly bring in more webinars. There are ones planned for the next week also. So stay tuned for that and stay safe. And as what Dr. Rattan has also emphasized, that take the vaccines as soon as possible. So till then, have a nice evening. Thank you, sir.
Thank you.
Thank you.